Okay, so now that we've learned about DNA structure and how it replicates, let's learn about some kind of more practical uses that is happening with DNA in today's society. So I'm sure you've heard of genetic engineering, but you might not really know what that means. And basically, it is us manipulating genes for practical purposes. So we've done this with agriculture, if you've heard of like GMO foods, right? We're doing this in research to see if we can like get rid of diseases or cure diseases. And we also do this in law to like do DNA fingerprinting to prove if someone did a crime or didn't do a crime. So, as you can imagine, it's pretty complicated to do this because in all of these cases, we are looking at just one gene, and chromosomes contain tons and tons of genes. So we're looking at an itty bitty little piece of a chromosome, right? And so it can be hard to zoom in and get that small and such a complicated polymer here, right? So we're just looking at one little chunk of it. And so because we're just looking at one chunk of it, if we're doing some like research on it, we don't just want one. We're going to need lots and lots of that one chunk, right, to run multiple trials, if that makes sense. So a big part of genetic engineering is going to be making copies of the gene you want to study. And that's called DNA cloning, is you're making copy after copy after copy. So you can continue to test different things to see if something's working or not working, or what if I do this? What if I change that? You need to have bunches and bunches of copies of genes, okay? So, okay, cool. Yeah. Let's get some copies of genes. How do I do that? The way that you get copies of genes is a process called PCR. And this comes from the word polymerase chain reaction. That's the P, the C, and the R. You're going to be seeing this in a simulation, so I'm not going to get too into the specifics, but basically is you take a segment of DNA, okay, and that has the gene on it, but has some other stuff too. Okay, but you take that segment of DNA and using different enzymes and using big temperature controls from getting it really, really cold and really, really hot, this is going to end up isolating the gene that you want. And then it is going to also, if we add in some more enzymes, it's going to copy that gene over and over and over again. Okay, and the specifics of that, you'll see the specifics in your simulation you do, but that's the overall idea. Okay. So at the end of a PCR, you get, billions of copies of the gene you wanted, but you also get kind of some border or buffer on either side. Because if it makes sense, if you want to study that gene, like you don't want to cut it too close just in case. So there's usually like bits on either end that is extraneous DNA that we don't actually want, okay? But if we're studying this one gene, all we want is that one gene. So at the end of PCR, you kind of got to do a little bit, another step, so that you can isolate just the gene and get rid of the bits on either end, okay? And the way that we do that is called gel electrophoresis. Again, you're going to be doing a simulation of this that will help you see this. But basically, you make this piece of like really, really thick science jello, okay? And you there's little holes in the jello that you can put your DNA into. And you're going to hook this up. Here's the electrophoresis part. You're going to hook this up to some electrodes and run some electricity through it. Now, maybe you're aware that when you've got electricity, you've got a positive source and a negative source, right? Like how batteries have those two different ends of the battery, okay? Well, DNA is negatively charged, so it's going to be repelled by the negative electrode, and it's going to go towards the positive, right? Because opposites attract, right? Sames repel. So that negative charged DNA is going to move through the gel towards the positive electrode. And but not all the DNA is going to travel at the same speed. The big chunks of DNA, which is that gene that you wanted to study, they're going to move slower because they're bigger. And the small chunks, the extra bits, are going to be able to move a lot faster. So they're going to travel a lot further. And so once you turn off your gel, once you turn off the electrodes, you can literally, like, this is your finished product. You can literally cut out like this top chunk, which has your gene, and then like you can just throw away this bottom chunk, which has the extra bits, and then you can go on and do your experiment with it. Yeah, with like these pieces of science jelly. Kind of weird. Here's what it looks like. Here's the science jelly. Here are the holes to put your DNA in. And then you can see it's hooked up to some electrodes here, and there's some like fluid in there to help it pass. Again, you'll be seeing this in a simulation, okay, but that's the idea, is it helps get rid of the extra bits of DNA that are left after the PCR. Okay, so now I've got my gene. How do I just go ahead and use this gene? And one of the most common ways we do that is we are going to put this gene into a plasmid. Now let's take a step back. Let's remind ourselves here, in prokaryotes, remember how they have like a circular chromosome? Prokaryotes often always have small little pieces of circular DNA that isn't part of the main chromosome. They're separate, and these little pieces of DNA are called plasmids. And they might have just a small number of genes, whereas this has all the genes. Okay, so if you can imagine, we got this plasmid here. We're going to take a specific enzyme called a restriction enzyme, and that restriction enzyme is going to cut the plasmid open. Okay, so here's our plasmid now. We're going to cut it open with an enzyme. Here's that gene that we had at the end of our PCR and our gel. And now we're going to put that gene in. We're going to get some ligase in there. Remember, ligase joins DNA molecules. We're going to get some ligase to glue it here and glue it here. And now we have this plasmid that now contains the gene that we wanted. Okay, and this is typically in a bacteria. So now the bacteria has the gene that we want to study. And now we can do experiments on the bacteria, and we can see how the bacteria responds, and that can help us figure out what's going on with our gene. Okay, so oftentimes we end up putting these into bacteria. Okay, that's our usual thing that we can do. And you're going to be doing this, actually. You're going to be doing some experiments on a bacteria, and you're going to take a gene that causes them to glow in the dark, and you, through the wonders of science, will be inserting that gene into this bacteria's plasmid, and if you do it right at the end, your bacteria will all glow in the dark. They'll glow bright green, which is pretty cool. Okay? One other way that we are using genetic engineering is through gene editing. Okay, and this is basically, if we study a gene, could we possibly fix a faulty one that might be causing a disease or disorder, or could we just turn it off entirely? Okay, and so we're finding now that we can actually do this, and one of the main things that does this is CRISPR. Maybe you've heard of CRISPR. We're going to read about it. And so CRISPR is an enzyme, and it is going to, let me show you. Okay, so CRISPR is going to make this guide RNA, okay, this purple bit. And this guide RNA is going to be made by CRISPR, and it's going to go to this very specific part of DNA. And then there's this accessory protein called Cas9. Here it's shown as this white cloud. And the Cas9 is going to go and find that guide DNA, that guide RNA. And it's going to attach onto the DNA where the guide RNA is. Here it is attached. And Cas9 is going to end up cutting this. Boom, snipping it right where it attaches on. And so this is going to cut both strands. Okay, so now we can either put in some DNA to help fix that gene, or we can just cut this gene out entirely and turn it off, which is pretty cool. Okay, so we now have that capability, which we didn't have before. It's a big source of research right now in science, is using genetic engineering to modify genes, and with CRISPR. So this might be a thing you might read about or hear about a lot in the news. So those are just some intros to some of the different fields of genetic engineering, and we will be talking about these or experiencing them in class. But here are the main highlights of what we covered in this lecture. And in the textbook, if you're wanting more info, more explanation, different pictures, paragraphs to read, you can check out 13.4.